So it's my pleasure to present a recent work from our group on exploring tumor-normal crosstalk with a new method, TranNet. And we use this method to explore the role of environment in tumor progression. This was mostly work by Bayarbaatar Amgalan, who couldn't come here, in collaboration with Chipping Day from Cancer Institute, and I am Teresa Przytycka. So there's a growing understanding that, so typically when we study the gene expression in tumor, it's often contrasted with the gene expression in the so-called normal control tissue, which often is being taken from these regions of the organ that's not too far from the tumor. So this is now, there's a growing understanding that this so-called control normal tissue, sometimes referred to as tumor adjacent normal, it's not quite normal. A recent study proposed that this, from Atul Bute group, proposed that this tissue is some in between normal and tumor. There are many studies recently that explore the tumor microenvironment that may also affect this tissue and its impact on tumor progression. And finally, many of the tumors are a result of some exposure to toxin, be that liver disease here or be that lung cancer. And therefore, if the tumor emerged as the response to toxic exposure, it's probably all the organ was also subject to this toxic exposure, so it's not completely normal. So why would like to understand this relation between the tumor and normal tissue? Whenever I say normal, in this case, I really mean normal adjacent to tumor. So if indeed is the case that this tissue represents some in-between phase between normal and tumor, it's interesting to understand what that phase may be and how that phase may be translating to tumor tissue. So that will help us to understand the tumor evolution. There's also important to keep in mind that that is quite well understood that, or appreciated, that tumor-related alteration of adjacent tissue are assumed to contribute to post-operative cancer recurrence. So the tumor is being removed, and nevertheless, it may recur in a couple of years. So again, understanding this tissue, this normal, presumably normal tissue would be helpful to understand this recurrences. And again, finally, if that adjacent tissue represents tumor microenvironment as well, there have been a lot of papers that suggested that tumor microenvironment modulate cancer progression. So we also would like to see how that information encoded in this normal tissue may be related to cancer progression. So because of this motivation, we decided to look at this relation between normal and tumor tissue by computing a transition function that will, given a gene expression in normal samples, compute a transition to the gene expression in target function in tumor samples. And given that that transition function, we would like to dive into what that transition function teaches us about biology. So let's start. Let me start from describing a little bit of the method. The main idea is to, as I said, is to compute a transition function from normal tissue to tumor tissue. And in this case, we have for every tumor tissue, we have matched normal. So we have a matrix describing gene expression in normal tissue and matrix describing gene expression in tumor. The genes whose expression we directly include in the matrix are the genes that are differentially expressed between tumor and normal. So we have G1 to Gn will be differentially expressed between tumor and normal. And then we also include principal components to represent the remaining genes. So the genes that did not make the threshold to be differentially expressed may still be informative, and I would like to understand how they may contribute to the transition. We do not take all the principal components, but we take the principal components that we call them differentially expressed. So what would be a differentially expressed principal component? So as I say, we have the samples from both blue would be tumor or gray would be normal, and for each sample we ignore all the genes that are differentially expressed and only keep the genes that are below the threshold, and we compute principal components for this set. And then the principal component will be differentially expressed. If you know along this axis, you can see differentiation between tumor and normal. So principal component 1 would be included because it will differentiate between tumor and normal. Principal component here, whatever that was probably 2, will not, because you know, it does not provide such a separation. So this is just so that we account for all the genes there. Okay, so now we know what are the two matrices, and what we do, we compute a transition matrix from X to Y, the transitional matrix M, that we can think of as a Bayarbaatar graph that goes from the genes in the normal tissue, genes and principal components in the normal tissues, to genes and principal component in tumor tissue. Now, additionally, we would like to pick up from the normal tissue, would like to pick up most informative genes, the genes that are most strongly predictive of the gene expression in the tumor tissue. So we enforce that by enforcing lasso constraint and enforce this sparse solution for this problem. That creates some challenging as how to find the optimal solution, and this I will not go into that. It can be found in the paper. Okay, so this optimization problem is being solved. How do we know we can trust this solution? So what sort of properties we would expect that will convince us that is interesting enough to dive into biology that is captured by this transition function. So we look at all the 3 elements of the transition, and for each of them we design some tests, some that will help us to convince that we should that the results are meaningful. For matrix X, the matrix that is representing normal tissue, we would like to argue that the genes that we picked up from this sparse selection process, which we also refer to as predictor, that those so-called predictors really predict gene expression. And the second thing, this is important since we use sparsification, lasso, that often leads to the solutions that may not necessarily be stable. So we have to somehow convince that at least in some regime that is most interesting, this is stable enough. For the edges, we will like to show that they are enriching functional interaction. And finally, for the genes that are going to be predicted, we would like to really show that they are predicted. So to do that, first we start with the validation of these predictors from normal tissue. So we rank this predictor according to what we define here as regulatory potential, or could be predictive potential also. So we call it regulatory potential. So this is simple, the sum of the absolute values of the weight of outgoing edges. If we sort all those predictors that have in this order, then we quickly observe that those predictors have kind of scale-free type of property, that most of them are really here in this little chapter would be 95%, have really small predictive power, and there is a relatively small fraction of them that have high regulatory potential based on this definition. So of course we would like to look at the ones that have high regulatory potential. So initially we arbitrarily say, let's take the top 30% of those genes and see how good predictive power those genes may have. So we do it by using leaf one out validation and comparing the prediction from the model that we get for the patient that is being left out to the prediction that we get by chance, and that give a very.
What are on the axis in that picture?
Oh, thank you. So this is so we have two things. One is the predictive the regulatory potential, and the second is the degree of the node.
Oh, okay.
Sorry, thank you for mentioning that. So of course you would expect that those two are related. That yeah. Thanks for this comment. Okay, so you know, the 30% is kind of arbitrary. So in the next step, we say, well, let's for every cancer that we consider in the study, let's find optimal cutoff. What would be the most predictive genes? And we simply do it by starting from the gene with the highest regulatory potential, keep adding, going along the list, and compute the t-statistic or p-value, and and we observe for all of those cancers that at some point we have obtained some optimal point, and then the predictability drops. It's still highly statistically significant. I didn't put the value here, but it's still highly statistically significant. But we were interested in this first group of them, and for individual cancer, up to the cutoff, we'll have like a small number, a couple of hundreds of predictors. So we decided that this is the predictors that we would like to test for stability. We do not expect that the ones that are not so highly predictive will be stable. They probably could be substituted by other genes, but we would like to confirm that the top predictors are stable. And basically what we do, we redefined what are, using different thresholds, what are the genes that are differentially expressed in cancer and normal, rerun the whole program, we obtain a new set of predictor, and we compute overlap. And you can appreciate that the overlap is really very high. This is the Jaccard index and the p-value of the overlap. So they are not identical, but they are highly stable. So this is so the predictor seems to be stable and interesting. Now, what about the target? Of course, we do not assume that you can take normal tissue and predict gene expression in cancer tissue exactly. If that was so obvious, then you know that would have been noticed long ago. But what we can observe, so this is here a distribution of the error, you know, that we have in the prediction, and you can appreciate that there's probably this big group that is not predicted well at all, and the tail that is relatively well predicted. And here, similarly, looking at the prediction from the R-square measure, again, it's not perfect. We don't have R-squared of 0.9, but most of them are more than 0.6, a lot of bigger than 0.7. So it predicts a good fraction of genes. It doesn't predict all of them. So, and finally, as for the edges, we use permutation test to confirm that they are enriched in human net functional edges. So that with those 3 points, we are confident enough that what we obtain is relatively stable, is informative. So we now dive and see what sort of genes are both in the set of the predictors and what sort of genes are being predicted. So the first thing which we do was kind of more like just looking at the top of the list. So for every genes, for every cancer analyzed here, we took top 10 genes in the set of the predictors, like sorted by this regulatory potential. And amazingly, almost all of them had supporting literature showing that those genes have been found as associated with cancer progression. And mind it, those are genes in normal tissue, so-called normal tissue. And just as an example, LYCK is a really well recognized and in many papers as a gene that is predictive of cancer progression. PRMT5 here is a gene that regulates the androgen receptor, androgen hormone. which is very important in prostate cancer. And our collaborator went through all this list and said they basically represent those three groups, either invasiveness or related to inflammation, or related for the ones that are in lung cancers, they're related to lung diseases, and also for the prostate cancer, there are some genes that are related to neurogenesis. So he was very excited about this last observation because there is now emerging notion that there is a relation between prostate cancer and neurogenesis. So we then look at, so that was manual, now we look a little bit more formally through the Go enrichment analysis, and this is just one table summarize like the main finding here. So we look at those cancers that we consider in the study. We look at the Go enrichment of the predictors, Go enrichment of the targets of those top 10 predictors from this table. And then also the Go enrichment for those guys that are here that are well estimated. So we can appreciate that for all of those cancers, some of the terms repeats. We have for the predictor, it's immune response, cell communication, and G protein coupled receptor signaling. So all of those terms, without exception, are related to features that are, or to processes that are external to the cell. So that are related to the environment. For the targets, also again, we have immune response, cell proliferation, tissue remodeling. So again, we have processes that are often being seen as a response to the environmental stimulus. And here we have one process that is characteristic to prostate cancer only, is this neurogenesis. For the last group, the ones that we just look at the enrichment of the top genes that are best predictor, again, we have similar processes. And in addition, for both of the lung cancer subtypes, we have enrichment in cilium. And you may be aware that the cilium function is very important in response to environment, in cleaning the mucus and cleaning the various toxins from the lung. And for liver cancer, again, you have one another very liver-specific term is detoxification. So again, that's consistent with an environmental impact on liver. So in summary, the approach TranNet provides a reliable method to study the interaction between tumor-normal and, as we found, mostly environment. Many predictors that we identify in normal tissue represent regulation, either directly or indirectly by the environment, as based on their functional annotation, and we have much more in the paper. We conclude that tumor growth and the genes that are, tumor genes are both that are regulated by those predictors relates to some are general, and there are also some are cancer specific, as you could see. And also, as for particular result on specific cancer, our results support the notion that tumor-normal crosstalk plays an important role in prostate cancer biology, so something that's being recognized. And I would like to acknowledge Bayarbaatar, who did most of this work, and the whole group, Chipping Day, who is our collaborators in the study and an expert in cancer biology. And I thank you for attention.
Are there any questions? Yes.
That's a nice talk.
Thank you.
So I didn't quite understand how do you define positive crosstalk and negative crosstalk?
Oh, so basically, so this is the matrix. So this is basically whether coefficient of matrix M is positive or negative. So basically, what we in maybe, I just wrote it as a matrix here, but basically what you want to is for any genes, you represent the expression of these genes as linear combinations of the genes that are in the normal tissue. So those are feature, and this is the expression of the gene we predict. We didn't measure. Most of them are positive, so most of those weight positive. So but it's possible that there are some repressors or something like that. So this is just the sign of the of the weight that is being taken for linear combination. We didn't really dive into this too much, but most of them are positive. Mm-hmm.
Thanks. Very nice talk.
Thank you.
I was wondering whether you've done some sort of negative control of taking, instead of the tumor proximate, take some completely different tissue.
We are. We are looking at this, but this will have to be done differently using with different approach, because here we are having matched tumor and normal. So we are going to take GTEx, but then we will not have matched tumor and normal. All right. So we have to. So we are looking at it actually right now and trying to figure out how we can have best of both.
Yeah, because sometimes in dimensions you can predict reasonably well also with random features.
Oh well, I mean, we did randomization. We compared. We did leave one out validation and compare. Whether the model predicts better than if you randomize the gene expression. And also, what also gives us the confidence is maybe something that may look not so good initially, but if you think about it, it's something that you would hope is that curve here, that you take the best predictor and they predict very well, and if you keep adding the predictors that didn't have such a high score, your prediction actually drops. So from that point, we probably have some overtraining, a lot of stability or something like that. So the fact that the top predictors we are getting the same independently how we defined differential gene expression, what is the threshold. So that's also quite encouraging. And then we often do manual validation. I've never have such a number of hits that almost everything we touch is supported by the literature.
Yeah, but there are differentially expressed genes that you take. So.
But those are identified in normal tissue, right? So that's, yes, they are differentially expressed because we don't want to pick up, say, sex or something like that. We wanted to protect ourselves from picking things that are unrelated to cancer. So yeah, but this is a good point.
One last question.
Sorry.
So great talk. I was wondering if you thought about extending this to include microbiome data, which turns out to be a good environmental surrogate.
That's a perfect idea. I don't know how much we have, how much, if we can collect the data from the same cancer types and things like that. Again, that will be the same as with the GTEx data. We'll have to forget about matched tumor normal, right? So we have to have some.
No, you can have matched. There are microbiome studies with matched tumor and adjacent normal.
Oh, okay.
So
That would be really
you
nice.
can even extract from the RNA-seq data as approximate. So that's really nice.
This sometimes doesn't work.
Right.
Because it goes through pathology, and
Okay.
pathologists are dirty.
What we just got. No, I know. We just got, this is a different topic.
Sorry. I've been on the record saying that.
I guess they've
No, but
accepted
this-
genome medicine to show that we do experiments really to experimentally validate our microbiome construction with a stringent type of data filter. You can get accuracy, so like matching but we can discuss this later. So,
Yeah.
No,
so
but
we
that's-
developed a pipeline for
that's-
this. So
Yeah, but you use some stuff too.
That's
we
very.
do some stuff, but as a proxy it seems to. So we do dish validation, 6 seconds, and
That's,
mnemonics to show that we.
Yeah, the positive signals are positive, but then you text it.
yeah, well, that's a great suggestion. So I just wanted to mention that the goal terms here, we just group them together and put some overarching term. They are much more detailed, and there's more biological information that can be digged from this term. This is just summary. Okay?
Great, then let's thank Teresa again.